Here, the remote jerk. Those are our friends, the grasshopper. They are everywhere, from meadows to rainforests. These fascinating insects play a vital role in our ecosystem. But how many grasshoppers truly share our world? Counting them all one by one seems like an impossible task. That's where science steps in with a clever method called Capture Mark Recapture, or CMR. So what is CMR? And why Orthoptera call this place home?
Grasshopper, Orthoptera, are the important players in our ecosystem. But estimating their population accurately can be tricky. But fear not, science have a trick. Today, we'll explore a clever technique called Capture Mark Recapture, CMR. This method allows us to spy on the hidden world of Orthoptera population that focus on the location of the area around Lake Alpha of the University Technology Mara Campus Tapah UiTM Tapah without having to count its insect. CMR involved to capturing samples of Orthoptera, marking them in the harmless way, then releasing them back into their habitat. Then, we capture another sample and see how many are recaptured. By analyzing this recapture rate, we can estimate the total population size of Orthoptera in the study area. This presentation will delve deeper into the fascinating world of CMR for Orthoptera. We will explore the steps involved, explanation of technique, tool use, and description of the Orthoptera species found. If the application of this method can contribute to the understanding of the diversity of Orthoptera insect species in terms of ecology, this helps us understand these fascinating insects and their role in the ecosystem. Now let's move to the objective of this experiment. There are several objectives that need to be focused on when conducting this experiment. First, estimate population size of Orthoptera. The main objective is to estimate the total number of Orthoptera insects living in the area of the Tasik Alpha UiTM Tapah. CMR provides a reliable method for population estimation without physically counting its insects. Second, contributing to the understanding of Orthoptera diversity. By identifying the species of Orthoptera cards, the experiment can contribute to a better understanding of the diversity of grasshopper, cricket, and related insects present in the UiTM Tapah area. Finally, require technique and tool for future research. The experiment can also serve as a platform to refine capture, tagging, and recapture technique used to Orthoptera study. This may involve testing different tagging methods or adjusting sample strategy to optimize the CMR process for future research effort.
Next part is the methodology. The method used in this study is capture and recapture technique. First, equipment. The equipment involves a sweep net. Second, technique. For the technique, the handle of the sweep are held firmly by both hands in order to have an accurate and fast sweep to capture insects. We also position our body at a suitable starting point within the habitat region. The third is sample collection. After a completed sweep, nets are quickly closed by securing it in order to prevent insects from escaping. And lastly, examine and release. The insects are captured, is observed, identified, and released back in the same place where it was caught.
So now, we will dive into the analysis of the data. We have found 3 species of Orthoptera in UiTM Tapah, which is Apida cinerea as species A, Akita domesticus as species B, and Calipera as species C. Based on the table here, we can see the most abundant species is species C, and the least abundant is species A. But before we are getting deep into the analysis, I would like to briefly explain to you guys about the characteristic of each species. First species is Acrida cinerea, commonly known as the oriental long-headed grasshopper, belong to the suborder of Califera. This species is recognized for its elongated body and long slender antenna. The coloration of Acrida cinerea can vary, typically presenting shades of green or brown, providing excellent camouflage among grasses and plants. Acrida cinerea prefer grassy area and open field environment that allow it to blend seamlessly with its surrounding. As a herbivore, this grasshopper feed on various types of grasses and other plants, using their powerful hind legs to jump away from predators and potential threats. Next is Akita domesticus, or the house cricket, belong to the suborder of Ensiphera. This species has a strong body, typically brown or greyish in color, with long antenna and large hind leg adapted for jumping. House cricket are commonly found in and around human dwellings such as home, barns, and other buildings. Unlike Acrida cinerea, house cricket are omnivorous, feeding on a wide range of organic material. Lastly, the suborder of Califera, encompassing various species of grasshopper and locust, includes species with shorter antenna compared to those in the Ensiphera suborder. These insects generally have a more robust body adapted for jumping, with coloration ranging from green to brown, often matching their habitat. Grasshoppers within this suborder are typically found in grassy field and agricultural area where they have ample food sources. Primarily herbivorous, they feed on grass and other plants. Now let's get back to the analysis. On the first day of capture, we found 3 species from species A, 4 from species B, and 5 from species C. All the specimens have been marked and released back into the environment. 14 days after the first capture, we are back to capture again, and we can see that the quantity of the captured specimen is decreasing to only 1 specimen from species A with 1 marked specimen found. For species B, we have found 2 specimens, and 1 of them is the marked specimen from the first capture. And finally, for specimen C, we found 3 specimens on the second capture, and 2 of them are the marked specimen. In this analysis, we employ the Lincoln-Peterson estimator to estimate the population size of the Orthoptera species in UiTM Tapah. Peterson estimator is given by the formula n equals to n times s divided by r, where n is the estimated population size, m is the number of individual capture and marked in the first capture, s is the total number of individual capture in the second capture, and r is the number of marked individual capture in the second capture. Through the application of the Lincoln-Peterson estimator, we have estimated the population size of the Orthoptera species in UiTM Tapah as follows, which Acrida cinerea estimated population size is 3 individual. For Akita domesticus, the population size is 8 individual, and Califera estimated population size also 8 individual. To improve the accuracy of these estimates, additional capture-recapture sessions should be conducted. Moreover, considering alternative estimator or method could provide more refined insight into the population dynamics of these species.
In our study, we successfully captured several Orthoptera over a 2-week period, focusing on their morning activity periods when they are most active in foraging, communicating, and reproducing. Our choice of expensive grassland as capture sites proved effective due to their prevalence as habitat for many Orthoptera species. Capture method involves simple traps made of netting and woods, strategically placed amidst tall grasses along the field edges. Our finding highlighted a diverse array of Orthoptera species, including grasshopper and crickets, distributed widely across our research area. In addition to capture, observation of their behavior provide insight into their unique social interactions and feeding patterns, depending on the type of vegetation around them. This study not only improved the understanding of the ecology of Orthoptera in their natural habitats, but also important to support their conservation and protection in their future. This discussion emphasized the importance of continuing further research to deepen our knowledge of the important role of Orthoptera in the ecosystem, as well as the steps that need to be taken to maintain species diversity in the long term.
And lastly, conclusions. In conclusions, we know that Orthoptera is an order of insects that comprises the grasshoppers, locusts, and crickets, including closely related insects such as the bush crickets, Tetidis, and Weta. Orthoptera have 2 varieties, which is Califera and Ensiphera. Based on our observations, we have captured grasshoppers and crickets, which is from Califera and Ensiphera species. Orthoptera have a generally cylindrical body with elongated hind legs and musculature adapted for jumping. Orthoptera mostly inhabits vegetation such as grass and shrubs, upon which they feed. To summarize, Orthoptera are animals known for their powerful hind legs adapted for jumping and stridulation, which is sound productions. Ecologically significant as herbivores and prey, they play roles in food chains and agricultures.